Okay, so let's just go to the next class. So in next class we see, and just to say that you can make this window bigger or smaller, and I'll just make it for the purposes of this, hopefully bigger. Come on. Okay. So another way of looking at that here is that that's the plot of these guys. So what we're looking at here is genes that are going up around 4 to 8, 10 hours. So these are our G1 proteins. So that you're there. If we assume that the cells are in G0 before the experiment, we add in the growth, and the cells are shooting up. They're coming up fairly early on, 2 to 4 hours after adding the serum. They're peaking, and then they're dropping down again. And you can see in the more time points we have here that this is a more gradual decline than in the where we only have 6-hour time points here, and we see it up at 6 hours, down at 12 hours. So this is just worth pointing out that actually, you know, there's often a need when you're doing a time course experiment. Kind of, we all want to do replicates. Replicates are good, but quite often when you're designing an experiment, you have to decide: do I want lots of replicates, or do I want lots of biological time points? A time point is a kind of replicate, because if you know that between 4 and 8 hours, if 6 doesn't sit in the middle of it, then there's something weird. So replicates can work. So when you actually put more time points, you get far more information about how it went up, when it peaked. When you look at only 6 hours, that could be the peak. It could be on its way down by 6 hours. You don't know, because you've got a big gap of time course where you don't really know any information about those genes. So generally I like to do lots of time points over lots of replicates. Yeah. And it turns out with this experiment, we learn nothing after 24 hours because everything kind of goes back to an asynchronous. Right, just wandering through this data here. Next class. So that's kind of quite similar. So let's just do this. So I can display multiple classes. Okay. So you see how similar those two are? Okay. Very similar in their overall expression. And what we'll find is, I'm pretty sure, is that when we look at the graph, they'll be in a similar place within the graph. So, see those purple nodes there and those brown nodes? Yeah. Two classes of which are the clustering has separated them apart, but when we look at the overall profile of those genes, they're extremely similar, and actually they're actually situated next to each other within the graph. Okay. What's also worth noting is that our cluster 1, which is over here, is very well separated from this cluster over here, these two clusters that we're looking at here. So that the graph is like a physical map to remember where everything is within this world. And this is a set of proteins here. Again, I didn't mention that, but if you put your mouse over any given node, it'll give you the profile of that specific node. Okay. Going back to the actual class viewer. Okay, we can I'll just wander through here. So here we have a set of genes which are going up later. So going up, peaking around 22 hours. They're interestingly quite high in the controls in that sample. Here's something that goes up very much around 12 hours, stonking big increase. And actually I know what these guys are. Where are they? Okay, see them there. They're kind of related to what was cluster 1, which was the going up at the biggest cluster. These ones are just sitting on the side, sitting proud. So they're kind of related to that, but they're not the same. One of the reasons that they're not the same is that you'll notice in this second thing is they go down. So these cells, when they go into proliferation, you only really see one. There's the idea that cells will go into cycle. They'll go into one cycle and then they'll go through a second cycle. What tends to happen with fibroblasts is that they go into the cycle and then it becomes an asynchronous population. This tells us that actually it's not completely asynchronous and there's a second division going on within this second 24-hour time point. It's only these genes here that show that. And if I reveal the identity of these genes, histones, tons of them. beautiful enrichment of histones there. You don't need a Go ontology to tell you what this list is and why they might be related. Okay, all the histone genes are going crazy. They're all captured. They have this quite specific pattern, which is different from a lot of the other cell cycle genes. Okay, and again, we'll just wander through here. Right, so now we're getting to a lot smaller list. So when you select a bunch of nodes, it'll say so how many nodes are selected here. So the real way that it's better to work with biolayout is to have two screens, one of which shows you the graph, the other one which shows you this. This way you can keep a visual record between what you see in terms of the profiles and where they are within the graph. But you can see these guys here. A lot of them are essentially not or quite up early on. Okay, and if we look at where they are within the graph, where are they? Did I deselect them when I did that? Okay, they are selected. I don't know where they are. So if you don't know where they are, one thing you can do, a quick trick, reverse the selection. So you've got some selected, you don't know where they are. So what we're going to do is reverse the selection. Okay, so that's now said everything that was selected, select everything else.
How did you do that?
So Control R. And then we can hide them and we can see where they are. There they are. Okay, so those were the genes. So to going back again, I can unhide them and I can see I've still unhid the no class nodes, which I'm going to re-hide. The genes we were looking at there were in here. Okay. Now let's have a look at some of these other clusters. So the other way you can look at clusters and graphs and what's in them is that you can say, well, why are these? What are this group here? So again, what you can do is select a node in there, say Control-Alt-S, and you'll have selected everything else within that, and you can then see what they are. So this is what I hope they would be. So this is a set of genes which are, and again, it shows you the power of time courses. By 6 hours, these genes, well, they peak here in the second experiment, but actually the reason that it only seems like a small bump is that actually these guys are up and down before 6 hours. Okay, so these guys are going up at 1 hour, maximum expression at 1 hour, and if we look at what they are, immediate early response gene 3, JUN-B, FOS, early growth response 2, early growth response 3. A lot of these are the early response genes. So in a lot of time course, if you look at the LPS data, you will often see if you've captured those early time points, these guys are changing. So these things are basically the first thing that change when we stimulate it. So let's just have a look at this graph in a kind of overall context, because it does kind of allow you to understand what you've got here. So we have all of these genes here. So these are genes, if we select some of these guys here, these genes are higher in the unstimulated cells. So at 0 hour, their expression level is highest. So these are things which are upregulated as cells go into quiescence. These are, if you like, your G0 proteins. These are things that keep cells from dividing. You'll find a lot of tumor suppressor genes within this collection of genes. Okay, so this is the set of genes which are upregulated early on. Well, they're not upregulated on. This is almost represents the stationary phase of cells. These are things which are specifically higher in cells which are in G0 than they are in cells which are about to go into proliferation. Then we see this event. Then within 1 hour, these guys are turned on. Okay. These are the early growth response. They are often sitting in open chromatin. They are turned on very early on. And what we're seeing then is then the increase in expression. So these ones here, if I look at them, will probably slightly later. Well, I'll look at both these two together. Okay. You can see one is shifted slightly away from the other. So the blue ones here are coming up slightly later than the orange ones here. But they're essentially all early response, just two different flavors. Often these genes are things which you actually have to look for because they don't have many friends. Because there isn't many of them, they're often not in that big cluster and you have to go looking for them because they easy to, it's a small cluster, I'm going to ignore it. But actually, they're some of the most interesting genes in terms of setting up the transcriptional profile that follows. Then you get into this phase here, which is G1. Okay, these are all the genes which are upregulated as cells grow and prepare to go into under division. So you're looking here at a lot of growth factors, a lot of metabolism upregulated here, and essentially this is the growth phase, and these will get turned off. And you notice how there's actually a thin web here. So there's a big burst of transcription. It then gets turned off before we go into what we're interested in, which is all of these guys, which are all the genes associated with the cell cycle, so the machinery of cell cycle. So within here, if you could break it down, it doesn't break it down very well as a graph, but here will be your S-phase proteins, so all your polymerases, all the proteins which are required for DNA copying, and then once the cells have copied their DNA, you'll see your G2 and M-phase proteins, which you can't really separate, coming up here, and your histones, which are part of that process, but are actually a bit weird because they go up and down. In their mRNA is not that stable, you see here. So what you've actually captured here is, we've done quite a simple experiment, but the answer is quite complicated, if you know what I mean. There's a lot of detail, a lot of interest here. Now, the thing for us that we've been following in our lab is that within this bunch of genes here, there's about 100 unknown proteins, okay? And we've been following them up and trying to prove that guilt by association actually real, and we've been knocking them down and doing protein localizations. And in fact, it turns out a lot of these unknown proteins do have all the characteristics of a cell cycle protein. Okay, so they're expressed in centromeres or other structures that we can characterize. You knock them down and the cells don't divide as well. So guilt by association, very powerful way for gene discovery and looking at this data. And you can hopefully see in this way how you can visually see the division of these genes into their kind of biological functional grouping. Is that reasonable? Just to say something about this data, this was not what it looked like when it came off the machines, okay? So when this machine, this is, the input for this is about 5,000 genes, and what we had to do then is that when we actually got the data from the machines, there were a number of technical issues with it. So as I mentioned, we ran this as 2 separate experiments. You will find in the raw data, well not the raw data, but the raw normalized data, that actually there's a difference between experiment 1 and experiment 2. There's a whole bunch of genes which are a lot higher in experiment 1 and lower in experiment 2 and vice versa. This is an artifact of normalization. These genes are not interesting. They're just, their expression pattern is changed purely because actually the data was different when we started. In that second experiment, which was ran on a series of 4 strip chips, one of the strip of chips worked differently. Again, there's a set of genes which are highly expressed in those 4 there. Now, the fact is that when we put this data in, first of all, you look at it here and you understand what the structure of the experiment was, and you understand that there's a whole lot of genes. It's nothing to do with the biology that we're looking at. And essentially what I have done then is gone through and removed those clusters. So I get rid of all the clusters which I'm not interested in. So the good thing and the bad thing about biolayout is it shows you the data warts and all, okay? I like that, but it does mean that it doesn't immediately, if you ask the question, what's the difference between 0 and 12 hours, you can do that statistically, you can get a list of genes and you feel happy about it. What you don't know is really what background you've selected those genes from and whether there was any technical issues associated with the comparisons that you want to do. So just to say a little bit about how we might do this. So I'm going to so this is a relatively simple experiment. There's 2 things to notice about this, that in time course experiments what we're actually dealing with is a continuum. So things go up and some goes up and some goes up, there's no, it's a series of stepped events, and what you'll often see in that kind of structure is an elongated graph structure, okay? So a bunch of genes which are all related to each other and have no relationship to anything else will form a tight cluster. So although you're not familiar with how these graphs look, when you start getting your eye in, you can almost understand the structure based on the way it's displayed. So here, this continuum shows this here. There's a decrease and then a burst here. So actually understanding that structure is quite important. And when you, most time course data, when Claire gets to look at her data, she'll see a cigar shape of LPS response, and that will represent it from the early to the late. One thing just to mention here that we can do, so we've implemented some quite nice animation. So if we hit this one here, what we're going to do now is go through each sample one by one, and we're going to make the nodes bigger or smaller according to the time point. So so you see this here. So this is the early response. You can just see, I'll slow that down. Make it about 0.5. So if you press this, this is the speed. How many entities, samples per second is it going to do? That's the node size that you're going to select. And what it does is essentially it makes the size of the node proportional to the maximum expression. So when the expression reaches the maximum, the node will be big and red, and when it's small, its expression is considerably less. So you can see, as I mentioned, this is the G0. We'll start seeing these guys going up. That's the early response, followed by the secondary response. Now we're getting into the actual main 6 to 8 hours. The cells have done their G0. We're seeing the cyclin E's coming on here. This is the S phase going on now. It'll switch over to G2M later on, I think. There we go. And then actually the second experiment will kick in. So 15, so we're on the 24 hours, and now we're back to control second experiment. Okay. So you can watch this nice response going off. Now you see we almost didn't see these guys light up the second time round because there was no 1 to 2 hour samples. So we went straight to the 6 hour event, and what you'll see after about 24 hours on here is you don't really see much happening up here. It'll get to a point and actually nothing much more is changing because the cells have become asynchronous, and essentially what you're seeing is the kind of combined mix of cells proliferating with those sort of just turning over. Okay. So it's interesting, actually probably these are more the G0 specific genes, because these guys are probably now silent. So it's quite a nice way of looking at the dynamics of data. Sometimes you can actually see stuff that way that you can't see by looking at it other ways. Works really well for time course data.